Pleasure to introduce our distinguished lecturer today, Dr. Agnel Sfeir, who's visiting us from Sloan Kettering. She attended the American University of Beirut for her undergraduate studies and then came to the United States when we've never let her go for good reason. She did her PhD at UT Southwestern, starting with what would become a lineage of superstars in the telomere biology field. And so she was, I think, co-mentored by Jerry Shea and Woodring Wright, the late Woodring Wright, unfortunately, because he's passed away. And actually, you know, I think before maybe joint mentorship was now this is much more popular, I feel like. But so you were an early pioneer. And the other thing about that marriage really, I think, brought cell biology because Ray was a big cell biologist. And that's always a part of Agnel's beautiful work. So we've known each other because after completing her PhD, Agnel then moved to Rockefeller to do her postdoc with Titi DeLonga. And so in the same building where I carried out my postdoc. So we overlapped for, I think, a few years there. And she taught me a lot, was my buddy at the telomere biology meeting, I think, at Cold Spring Harbor. And then she went on to launch her own lab in 2012 at the Skirball Institute at NYU. And then has moved back uptown again to the old haunts at Sloan Kettering in 2021. I think, I don't think you've been here since, I was looking it up, I think 2016. So at a 2016 visit to MCDB, I remember this amazing story of this new area that I was going into in mitochondrial DNA. I think it was kind of a new project that had started in the lab and now has just grown tremendously. And I think we're going to get to hear about this crosstalk between nuclear genome and mitochondrial genome, which is a really innovative and super exciting area. You know, she has, you know, won many prestigious awards, including the New Innovator Award from the NIH and the Pershing Square Sun Prize, many others. And it's my distinct pleasure to have you here today. And I'm going to first bequeath on you. Oh, it's lovely. We're going to get a picture. Oh. Well, maybe I'm going to have somebody take our picture. So can somebody volunteer? Thank you. Wow.
With my lab.
Yeah.
Thank you.
Okay.
Thank you so much.
Thank you so much.
Thank you, Megan, for the invitation and for this super kind introduction. It's always nice to visit places where you're invited by people you did postdocs around the same time with. And so as Megan mentioned, my lab is interested in understanding genome stability, which is very much rooted in my training. And when I talk about genome, it's typically both genomes, the nuclear and the mitochondrial. And so what I'm going to tell you today is two stories. The first half will be exclusively dedicated to nuclear DNA repair, which is a major area in my lab. And then the second half, I'll go into this new area, which is not so new anymore in my lab, but it's half of my lab working on it, which is mitochondrial DNA. And before I start, I do have to acknowledge these folks. They are the people who do all the work. My job is easy because my job is to make their life difficult. So, and I'll try to acknowledge people as we go through. And before I go, I also want to acknowledge my conflict with Repair Therapeutics, a company I helped co-found. So let's start with DNA repair, double-strand break repair, which probably doesn't need much of an introduction to many of you in this audience. But the most toxic lesions inflicted on our DNA are double-stranded break. There's nothing more dangerous than chopping your chromosome in two. And our cells evolved multiple pathways to deal with this damage. The safest pathway is homologous recombination. This is a pathway that relies on the BRCA1, BRCA2 genes, which, if you know Ryan, you know these genes. And This is a pathway that basically uses the sister chromatid to fix these breaks and therefore is error-free. But you need a sister chromatid and therefore you're confined to SNG2 phases of the cell cycle when there is a sister chromatid. Now, most cells in our body are in G1 and they rely on canonical end joining to repair their breaks, now, which is a stitching mechanism. The pathway I'm interested in the lab, the pathway we spent 10 years working on, is called microhomology-mediated end joining. This pathway is also known as alternative end joining, backup end joining, theta-mediated end joining. There's actually more names for this pathway than the actual genes we know of that pathway. But it's simple, it's mutagenic, it relies on microhomology. So an A, a T needs to anneal in order to joining to happen. It is a joining reaction relying on ligase 3. Now, a few years back, my lab identified a key enzyme in this pathway, which is polymerase theta. And I and I'm going to tell you a little bit about that. But to put MMEJ a little bit in perspective, this is a pathway that's found from bacteria all the way to humans. Even if Pol theta, which is coded by the gene PolQ, is not found in E. coli or lower eukaryotes cerevisiae. But the footprints of MMEJ are all over. It was first identified as a pathway that repairs the break left from a transposable element when it pops out in flies by the classical work of Mitch McVeigh. Then it was found to be important for integration of DNA, whether plasmid DNA, tDNA, or transposable element in some cases into our genome. If you want to mutate a gene using CRISPR-Cas9, you're actually praying that MMEJ is working because this is what's going to create the indel that will mess up your gene. MMEJ is also involved in some cases in dealing with replication blocks that HR cannot deal with, and it's responsible for chromosomal translocations. Okay, so we stumbled on this pathway completely serendipitously. I had no idea what MMEJ is or what Pol theta is, and I was working on telomeres, which is where I got trained at. And we had these cells that were a bit crazy in the sense all their telomeres fused to each other, creating this spaghetti-like appearance. So I went ahead and maybe the second or third experiment we did when we started in the lab was to sequence these cells, asking what's at the junction of two fused telomeres. Telomeres are Bolling repeats, TTAGG, AATCC. And so we were wondering what's at the junction when two telomeres fuse. The naive in me was thinking it's going to be a unique base of the 6 base pair repeat that I can then make a TALEN or a CRISPR and cleave it. Instead, what we got were random nucleotides that matched nowhere in the genome, and they were obviously not telomere sequence that somehow made their way into two telomeres when they're fusing. A small-scale shRNA screen of known polymerases that are low fidelity gave us one and only one hit, polymerase theta, which is coded by the PolQ gene. When you deplete it, you get a significant reduction of an end-to-end fusion. You abolish any sort of indels at the MMEJ. The key moment or the key finding in this paper was the idea that what we noticed is that there is rewiring of double-strand break repair. What I mean by that is that cells that cannot undergo HR, cells that are deficient for BRCA1 and BRCA2, somehow shunt or shuttle their repair into MMEJ, becoming an essential pathway in BRCA1, BRCA2 deficient tumors. And that drives synthetic lethality when you delete PolQ gene in BRCA1, BRCA2, PALP2, or other cancer mutated lines. This obviously had a profound clinical implication for many reasons. The one which made me convinced that we should go after it in the clinic is that POQ is not expressed in almost all tissues in our body, but is highly expressed in tumors. It isn't expressed in some T cells, it's expressed in the germline, but this strict lack of expression makes me believe that it's going to be any drug is going to be tolerable. And right now, there's more than 8 inhibitors that's being developed. There's 4 of them, 4 drugs that made it into clinical trials, including one drug that I helped developed with Repair Therapeutics, which is RP3467, which is the clinical trial is called Polar. It's being, patients are being recruited all over the US and Europe and at MSK. And they're being, these drugs are being tested in HR defective tumors, obviously, as a monotherapy, but also as combination with PARP inhibitors, which is the standard of care for breast ovarian tumors. And I'm showing you one graph, which is an in vivo experiment. This is BRCA2 deficient human cells injected in the flank in the mouse, treated with either PARP inhibitor alone, POQ inhibitor alone, or the combination, to show how profound synergistic effect we see when both drugs are combined. And even when you stop the treatment, it seems we're not getting reversion or we're not getting relapse of the tumor. But ultimately, right now, these drugs are the clinic. This is the ultimate experiment, and time will tell us if they're going to show efficacy or not. What I'm going to spend my time on is not the clinic, but actually the fundamental basic science, which is what we're good at in the lab. And to probe really deep questions about what is MMEJ? What are the genes in this pathway? And minimally, what is the raison d'etre? What I mean by that, if you think about HR, you quickly think about meiosis. You need HR to shuffle the genetic material. If you think about NHEJ, you think about antibody diversity, VDJ class switch. Why would nature evolve a pathway that's mutagenic? There must be a function for it. That's not just the backup that we don't know about. And Ali Brambati, a postdoc who used to be in my lab, now runs her own lab at the University of Colorado, she thought, you know what, if I want to ask the how and the why, I need to first understand what's the genetic makeup of a pathway. Once I know all the genes, then I can probe how and why. And she wanted to set up a genetic screen to identify all genes that are in MMEJ. And she thought, okay, it's simple. If I make cells that cannot undergo canonical end joining or HR, these cells have nothing to survive but MMEJ, and I can run a simple synthetic lethal survival screen to identify genes that are required for the survival of these cells and therefore possibly MMEJ factors. So she carried out a genome-wide CRISPR-Cas9 screen using either cells that are p53 null or cells that are BRCA2 ligase 4 and p53 null. We had to delete p53 when you block HR or NHJ, otherwise these cells are just so sick, they don't grow. And then she infected these cells with a library of 30,000 guides, and she did a classical survival screen. You take time point 0, time point 14, you sequence the DNA, and you identify the guides that are depleted in this setting, not in this setting. And these are guides that are essential only in cells that cannot undergo HR and NHJ and possibly MMEJ factors. The screen worked beautifully because basically what I'm showing you here is the base factor score, which is an essentiality score. So the higher the score, the more essential the gene is. This is the p53 and this is the triple knockout. This is the quadrant that we'd care about. We got all the usual suspects. We got PORQ, we got things that are known to be synthetic lethal with BRCA, APEX2, and others. And we got 3 members of a 3 subunit complex called 911 and their interacting partner, Rhino. I don't know if you've run genetic screens, but when you run a genetic screen and you get 3 subunits of a 3 complex, subunit complex, it's just easy where to go. You're not going to be validating a lot of hits to go after. So Ali decided to focus completely on this 911 complex and its obligate partner, Rhino. And the 911 complex was actually nice because this was around the time I moved to MSK. And the structure of the 911 complex was solved by Richard Haidt at MSK together with Dirk Remus, my colleagues. And so we could just go down to the 9th floor and ask them all the questions. Okay, and 911 is actually, it's a clamp. It clamps around DNA at the single-stranded, double-stranded junction. So the first thing, and what we knew about 911 from the literature, and that's 99.9% of the literature, is that 911 is involved in activating ATR kinase, which is the S-phase kinase. And so our first hypothesis is potentially the role of 911 and Rhino in MMEJ is by promoting signaling. Repair needs signaling. When you blunt signaling, no repair, right? It wasn't that simple because it turned out not to be the case. If you deplete ATR or any other ATR activator or downstream factors, MMEJ still took place even if ATR signaling was shut down. That quickly told us 911 is doing something different for MMEJ that's not through its canonical ATR signaling function. And when you have a clamp loading around DNA, and the 911 is often called the cousin of pCNA, the first thing that comes to mind, if pCNA is what recruits, the platform that recruits polymerases to the DNA, could 911 be actually the recruiter of polymerase theta to perform MMEJ? If it recruits, needs to interact, right? So we spent, I didn't do any of that. Olivia and my lab spent some while trying to detect a direct interaction between the 911 complex, Rhino, and pol theta. And I'm going to make a long story short, 6 months worth of work. We could only detect a robust, reproducible interaction between Rhino and PolQ. We could not detect interaction between 911 and PolQ. Now, we could detect this interaction if we do a co-IP by overexpressing proteins, by stably tagging endogenous proteins, using antibodies, or in vitro reconstitution. And I'm showing you one example without damage, without IR. You pull down MYC for Rhino and you see PO-theta binding. We did not see this interaction with RAD9, HUS1, or RAD1. And that was a puzzle for a while. And at some point, I told Olivia, stop. Let's just declare Rhino is what interacts with POQ and let experiments in the future help us solve that riddle. And so basically, Rhino seems to be that factor within the 911 complex that binds PO-theta. And I thought Olivia was going to drop it, but she didn't. And she came to my office like 2 months later to say, I think I know what's wrong. She had done a simple experiment, which is not sequencing, not databases, no, nothing. It's the simple experiment is a Western blot that many people nowadays don't know how to do. And that Western blot was on Rhino 911 and controls in cells that were synchronized at the G2M and released into M. And then Olivia took a sample after a few minutes and ran a Western. It was clear that Rhino is only stable in M phase. It's degraded upon mitotic exit. It's degraded because it has a CAN-box, T-box, which are cleaved by the APCC cleavage complex. Things now started making sense because Rhino only interacted with PolQ when it's expressing, which is in M phase, and we could see this interaction, not an interphase. Also started now, we make sense why we could not detect the interaction between 911 and PolQ, because when we were pulling down 911, we were pulling down the 99% of the protein, which is not in mitosis, and during that time, there's no PolQ in that complex. But now if we synchronize cells in mitosis, we pull down 911, we see everything coming down, including PolQ and Rhino. Okay. Now, so that basically raised a conundrum. If Rhino is an MMEJ factor, if Rhino interacts with PolQ only in mitosis where it's stable, and if PolQ is an MMEJ factor, what the heck is going on? Is MMEJ a pathway that's acting in mitosis, right? So what do we know about double-strand break repair in mitosis? We knew quite a bit. So we've known from the 50s that if you make breaks, if you irradiate cells in mitosis, they are susceptible to damage. And it's But HR and NHEJ are completely suppressed in mitosis. You do not do HR, you do not do NHEJ. And the prevailing idea has been, well, mitosis is short and quick. You don't care to fix breaks in mitosis. If you have a double-stranded break, hold it together, tether it together by the CYP2A MDC1 TOPP1 complex until cells get to G1, and therefore in G1, NHEJ can deal with that break. I did not like this reasoning because the last thing cells want to do before they divide is have a floating chromosome that is somehow loosely connected to each other, right? But there has been snippets of evidence from the literature that there is a function of pol theta in mitosis. Work from Marcos Lobridge, David Pellman, and others. So Ale decided to do the killer experiment, which basically synchronized cells in mitosis, make the damage in mitosis, take away MMEJ in mitosis, and ask, do these breaks get repaired, and are they dependent on MMEJ? So she basically took cells that are either POQ plus or minus, synchronized them at the G2M, released them into mitosis with no cortisol just to elongate mitosis in order to work with it. And she gave them 2 gray of irradiation. And she harvested cells at 1 and 5 hours post-irradiation and did a gamma H2X IF as a proxy for the lack of repair. So the more breaks, the more gamma H2X. And so in wild-type cells, you get a boost of damage an hour post-irradiated, and there it's all gone, suggesting there is double-strand break repair in mitosis. You are resolving MH2X, and you can only resolve that if you repair the break. And if you do the same experiment in a POQ deficient setting or cells that have a polymerase mutant in POQ, you do not get repair, saying MMEJ is the major repair pathway in mitosis. It's not a backup for HR, it's not a backup for NHEJ. Both of them are shut down, and this is the primary repair pathway in M phase. We get the same results if we treat the cells with POQ inhibitor in M phase, if we delete rhino, or if we use a rhino mutant that doesn't bind POQ, okay? Now, I want to go back, though, to this clinically relevant experiment, because first of all, reviewers said, how often do you get that many irradiation breaks in mitosis? Mitosis is short. Typically, the breaks happen in S phase, and if they're not fixed because checkpoints are messed up, they end up in mitosis. So we wanted to do an experiment that would model more this S phase damage that's happening but not being repaired, but spooling into M phase unrepaired. And so we did this experiment where we took cells that are either treated with siRNA control or BRCA2 depleted to block repair in S and G2, block HR. And we synchronized these cells in such a way that we treated them with PARP inhibitor in S phase, let them go to G2, remove the PARP inhibitor, and when they entered M phase, we gave them POQ inhibitor, and then we harvested the cells in M phase, and we did an IF for H2AX. Okay, let's go through the graph one step at a time. If the cells are wild type for HR, they're wild type for BRCA2, it does not matter what you do to them. They're going to fix the break in M phase equally to control, whether you treat with PARP inhibitor, POQ inhibitor, or combination. It does not matter. HR deals with business in S phase. If you cannot do HR in S phase, you start automatically seeing more DNA damage, more H2X foci in M phase without any insult. This is more in cells treated with PARP inhibitor in S phase. This is more if you block MMEJ in mitosis, and the combination of these two is synergistic. This is starting to explain to me how is this synthetic lethality happening. My idea would be, if you are BRCA2 deficient, you cannot fix damage in SNG2. Now you become reliant on M phase to fix the damage by MMEJ. When you add an insult to this, which is PARP inhibitor, and you can't fix it by BRCA2, you get this profound synergy because MMEJ is acting in mitosis. And in fact, we did control experiment. If you move this up here and you combine it with PARP, you do not get the synergy. The synergy needs to be when POLQ inhibitors are added in M phase. So that brings me to the model, which is quite simple. G1 is for NHJ, SG2 is for HR, and MMEJ is the major and the primary repair pathway in mitosis. You get this restriction on MMEJ at two levels. First, rhino, which is a key MMEJ factor, is only stable in mitosis and degraded upon mitotic entry. And second data I didn't show you, there's a PLK1 phosphorylation of rhino that is required to recruit PORQ to DNA damage sites. And so with this two level of control, you ensure that this highly mutagenic pathway of repair can only access the chromosomes in mitosis, right? So it's a last-ditch pathway. When chromosomes get to mitosis without being repaired, you bring in this mutagenic pathway, which is better than doing cell division with unrepaired DNA. So that brings the interesting question now. Is this M phase kind of honing or confining MMEJ to M phase, is this beneficial? Is this how you tame the mutagenic effect? One thing we're doing to test this idea would be to unleash rhino and PORQ through all stages of the cell cycle, boost MMEJ activity high, and see if it's going to start messing up with NHJ and HR. And are we going to see more footprints of MMEJ activity, which is the endas and microhomology? And this experiment is ongoing. Okay, where are we going next with this? I told you a little bit about, in the beginning, about MMEJ being involved in this integration of tDNA, circular DNA, transposable element. And there has been some snippets of evidence about MMEJ role in regulating microDNA. And that led David Billing, a radiation oncology fellow in my lab, who's absolutely brilliant, that led him to think, is MMEJ actually involved at all in maintenance biogenesis of extrachromosomal circular DNA? So, you know, ecDNA were identified in the 60s as double minutes, and with the advent of sequencing, just like everything else, they were re-identified again, rediscovered, and they're called ecDNA, and they're actually found, and they're prevalent in lots of tumors. They're multiple cancers, they're typically associated with poor clinical outcomes, and they basically provide the genetic heterogeneity that drug resistance thrives on. Now, David did one simple experiment that convinced me that he should keep going. He took cells, which are colo-DM. They have an amplification of MYC on an ecDNA, and counterparts, which are colo-HSR, which I'm not showing you here. And with those cell line, you can easily identify the extrachromosomal DNA by doing a FISH for MYC on a metaphase spread, and you see it outside the chromosome. And he treated these cells with PORQ inhibitors for a week. And he used two different types of inhibitors, a polymerase and a helicase. And so he ran a metaphase spread and counted the extrachromosomal DNA. It was clear. If you block MMEJ, these cells have HR, these cells have NHJ. If you block MMEJ, you get a reduction in ecDNA levels after a week. He sees the same thing if he makes a rhino knockout. And the interesting thing, these ecDNA are never completely wiped out. They go down like a tenfold, and they're sticking around. You don't see any of this if you block HR. You don't see any of this if you block NHJ. So somehow, MMEJ is required to repair and maintain ecDNA. But where is it acting through the ecDNA life cycle? So you can imagine ecDNA needs to form, they need to replicate, they must segregate, and once they segregate, if they have damage, something needs to be repaired them, otherwise they're going to be eliminated or integrated into the genome, right? So we know ecDNA is not involved in the formation, MMEJ is not involved in the formation of ecDNA, and we did some experiments to rule this out. We are still testing the replication part, but we ruled out also that it's segregation. It's not segregation. We study ecDNA segregation upon inhibition. Nothing much changes. But what we're now zoning in is this idea that the circular topology combined with high transcription rate of the ecDNA creates a lesion that needs MMEJ for repair. And when MMEJ is not there, these actually end up being in micronuclei and they get degraded. And I say this is some preliminary evidence. If we take these colo-DM and colo-HSR cells and we treat them with POLQ inhibitor, we start seeing accumulation of micronuclei only when we block POLQ in the colo-DM, and these micronuclei are all positive for MYC. Okay, there's a lot to do here. So we know it's MMEJ activity, not HR or NHJ, that's required for maintenance of ecDNA. If you inhibit MMEJ, ecDNA ends up in micronuclei and then is degraded. And we believe, or our hypothesis is that this function is consistent, is maybe MMEJ fixing S-phase damage in mitosis, and that S-phase damage is coming from a replication transcription conflict. And that's our work, like hypothesis that we're testing and trying to refute now. So what's the source of ecDNA damage? Potentially transcription replication conflict. And is there any functional consequence for this? Do cells care if they have less ecDNA? And the answer is, I think they do, because David did one experiment where he took HeLa cells that have an amplification of the HFR gene on an ecDNA, and the presence of the HFR on an ecDNA makes these cells resistant to methotrexate drug. So he took these cells, treated them with POLQ inhibitor, and then added methotrexate and POLQ inhibitor. And you can appreciate from this graph here that these cells grow much less, suggesting that you need, if the cell needs an ecDNA to survive, blocking POLQ can lead to the cell death. Okay, I want to summarize this part by talking a little bit in terms of evolution, right? So going back to this question of what is the evolutionary trajectory of MMEJ? On one hand, its function in mitosis suggests that maybe it evolved as a pathway to deal with damage that HR cannot fix in S phase. You can imagine if you have a break where you have a G quadruplex or a secondary structure that makes this chromatid unavailable to serve as a template, then you're stuck. You cannot fix that break, and therefore you maybe need a last-ditch pathway in mitosis, which is mutagenic, who cares, but fix the break. Alternatively, this increasing evidence from the literature about the role of ecDNA in transposable element integration of tDNA, microDNA, and ecDNA kind of allows me to hypothesize maybe this pathway is a gatekeeper that protects linear chromosomes from invading extrachromosomal DNA, viruses, endogenous and exogenous. So basically, it's the gatekeeper. You know, genetic diversity, genetic evolution needs some of those to go in, but a lot of them will create genetic havoc, and that's not good. So how do you control? Well, you put someone by the door to open the door slightly and control how much you open it, and that could be the MMEJ pathway. What's nice about these evolutionary ideas that it's going to be difficult to test them, but they're out there and we're still working on them for years to come. All right, I can take few questions because before I shift into the part on mitochondrial DNA, if you want. What do you think, Megan? Yeah. Yeah, so actually you get a lot of synthesis, profound lethality with NHJ on its own. So a cool ligase 4 DNA PK are lethal with POQ. And this is not my work, our work, this is the work of Dale Ramsden. And you see profound synergy with Fanconi pathway, as Alan DeAndrea said, like FANCD2, FANCI are synthetic lethal with POQ. FANCD2 could be part of HR. And it's very interesting. What is the basis of the lethality between NHEJ and MMEJ? That's a riddle we're trying to solve. Is it rhino independent or is it rhino dependent? We're trying to figure this out. Yep.
About the polymerase and HPV, because it could become integrated during cancer, but it maintained extrachromosomal during regular infection. What is the role of that?
Yeah, so this is also not our work. This is work from Simon Powell and his colleagues at MSK. In fact, HPV, when it integrates at its junction, there's microhomology, and that integration is driven by MMEJ. So, and the question would be, could you use these small molecule inhibitors to block HPV integration? This is, they're trying to look at that. Yeah. Yeah, so in the germline, actually, which is interesting, in the germline, POLQ levels are high and then they're shut down upon differentiation. And in there are some bone marrow cells that have POLQ, and I don't know if this is just a boost in mitosis. It could be. But I was actually thinking, trying to ask, POLQ high expression correlates with what? And that's not like what other gene, right? And it actually correlates with activation transposable elements, which brings this to me back. Like it seems that this pathway somehow if co-evolved maybe, or it's co-expressed whenever transposable elements expression is high, as maybe to tame them as well. Okay. All right, so I'll switch gears completely and get out of the nucleus into the mitochondria, something I've been fascinated by for a while. And, you know, I don't know, for the trainees, I started my lab after working on telomeres for 10 years. And I thought at some point, I'm too telocentric. I should just try to do something else. And I spent a month reading non-telomere literature, and I got sucked into the world of mitochondrial DNA. And I know Jerry Shader used to be in Yale, and I came to his office in, I think, 2014, and I told him, I need to learn from you. And he was incredibly helpful and incredibly generous with his time. Okay, why I care about mitochondrial DNA? The problem is simple, right? A cell has two genomes in two different organelles that need to function together. Otherwise, the cell is going to be dysfunctional. All right. Now, this genome is tiny. It's a 16 kb plasmid that codes only for OXPHOS subunits and some tRNA. This mitochondria has 1,500 proteins, all made in the nucleus, translated in the cytoplasm, delivered to the mitochondria. So they need to crosstalk, okay? Now, this genome is very interesting. There's no introns. It's all exonic. It's transcribed into one large polycistronic RNA that's cleaved into smaller RNA subunits, translated in the matrix by tRNAs and ribosomal RNA that encoded here, and then docked into the OXPHOS. Now, the rest of the OXPHOS subunit are made in the nucleus, and they're docked around this. Now, this is subject to so many different forms of mutations. And from point mutations, double-strand breaks, deletions, and depletions. So in my lab, what I've been doing is trying to create a tool to mimic all of these mutations and simply ask what happens. What I'm going to focus on today is deletions. Deletions in mitochondrial DNA, there's 120 of them that are identified in patients. And they're picked up and they range between less than 2 kb, where you delete a couple of genes, to more than 5 kb, which is like more than a third of the genome is wiped out. They're identified primarily in these classical mitochondrial pathologies, Kern-Sire, PEO, Pearson marrow, and they also accumulate in aging. And there's some interesting features of them. They're heteroplasmic in nature. What I mean by that, mitochondria is not just 2 copy, it can be up to 1,000 copy. And these mtDNA deletions do not take over 100%. They're anywhere between 0 and 60% or so, right? You need to have a little bit of wild-type genomes around, otherwise the cells are fried. They primarily impact muscle and brain, and are like tissues with energy demand. Why though? We do not know. And they're selected against in many mitotic cells, and they're not inherited. So the germline sheds these mutations. There's no cure. If a patient shows up with a deletion, actually the first manifestation, how you prognose a patient with mtDNA deletion, this muscle of the eyelids shuts down completely because that's the muscle in the body that we use the most. And that's the one that has the highest energy demand. And so when the patients become blind because the muscle shuts down, that's how you diagnose them. There's no cure. You only give them some painkillers and stuff like that to keep them going. Now, the most widespread deletion is called the common deletion. It's a 4.9 kb deletion that's flanked by 2 repeats. And how the deletions form is actually a replication slippage mechanism. So basically, you start replicating the DNA, something slows out the polymerase, the helicase keeps unwinding, you uncover repeats, these repeats misanneal to each other, you end up losing a piece of DNA. This, though, is incredibly, incredibly rare. Now, there's so many things we don't know about the deletion. Like I said, what's the threshold that triggers mitochondrial dysfunction? We can't get this answer from the patient. Some of them are sick when they have 40% of the mitochondrial deletion. Others are sick when they are at 70. And why some cell types accumulate deletion, others don't, right? Why doesn't the muscle or the neuron, if they need the energy, why don't they shed the deletion, right? And minimally, how does the cell respond to the presence of a deleted mtDNA genome in its mitochondria? Now, to address these questions, you need models. And the problem is there's limited cellular genetic tools to make the deletions and study them. Right? There's a number of challenges. First of all, you can't do gene targeting in the mitochondria. What I mean by that, the inner mitochondrial membrane is negatively charged, so you never can send a DNA or RNA, so CRISPR does not work. But we can cheat. We can use some archaic methods like restriction enzymes, talons or zinc fingers, and send them to the mitochondria, and we get around making a break. The other challenge to making a deletion is that there's no repair. There's no double-strand break repair. So typically, if you make how you make a deletion, you cut around a region and you want end joining to join the rest together, right? There's no double-strand break repair in the mitochondria. So you make a break and the DNA gets wiped out by nucleases. The MGME1 at twinkle, it's completely degraded. And you can see this here. If you express a restriction enzyme that cuts in one region and you measure copy number, mtDNA is depleted. The few molecules that are lucky enough not to be cut then repopulate and go back up. Okay, so Ifu, a grad student in my lab, thought if we want to make deletions to study them, I need to solve the issue of the lack of repair. I can make breaks easily. I need to put end joining. We cannot put human end joining in the mitochondria. It's too complex, many subunits. So we went and found the simplest end joining mechanism we can find on Earth, and that is from Mycobacterium, because it's 2 components, cool ligase D. They do the trick in vitro and vivo alone. And he thought, that's easy. I can express these 2 subunits, put a mitochondrial localization signal on them after a human codon, optimize them, express them in human cells, and boom, they go to the mitochondria, and they both of them, and they actually work nicely. So what I'm showing you here in this plot, these are cells which are RPE cells that are either expressing cool ligase D or wild type, and they got a DOX-inducible SCA1 that cleaves 4 times in the major arc. So the farthest one are 3.9 kb apart. And then we took these cells, we expressed DOX for like 48 hours. They're constitutively expressing cool ligase D, and we run an attack-seq. So mitochondria doesn't have nucleosome, right? So it is the contaminant when you run attack-seq. You typically want to trash it, but we love it. And attack-seq is the cheapest way to sequence mitochondrial DNA. So we run it all the time. And you can see if we analyze these reads, you get deletions, which are marked by blue, in 30% of the genome, and 800 different type of deletions, only when cool ligase D is expressed in the presence of SCA1. So we basically were able to reconstitute end joining and make mtDNA deletions in these cells. Now, if you've noticed, that these are the farthest SCA1, but the deletions extend way beyond the SCA1. This is because Ku, no matter how efficient it is, it's still fighting with nucleases, and it's not great at doing that. So if we block the resecting nucleases, MGME1 or polgamma exoactivity, you get way more defined deletions. This is good because now we can make any deletion, large, small, patient-associated, in any cell, epithelial, IPS, and we're actually now differentiating these IPS into muscles and neurons. And we can start, this is where the fun starts, because now we can start, what do these deletions do? Why they're different here to here, and so on and so forth. What I'm going to focus on the last 5 to 10 minutes is to tell you our initial work on this large-scale deletion, which is a SCA1, a 3.9 kb, in retinal pigmental epithelial cells. If you delete this region, you're basically removing subunits of complex 1, 5, and 4, and a bunch of tRNA. So that is very much similar to these large-scale deletions in patients. And you only accumulate it when both cool ligase D are present in the presence of SCA1. Now, one thing we faced, which we thought was an issue, is we got heterogeneity. Now, you don't want to let me distinguish between heterogeneity and heteroplasmy. Heteroplasmy is the number of wild-type DNA, mutant DNA versus wild-type DNA per cell. Heterogeneity is among cells. So one cell had 1% of the DNA had the deletion, another had 70%. So to solve this issue, E did a simple experiment where she basically isolated or derived clonal cells from that population. And each clone cell was characterized, and it had one level of heteroplasmy. And she created a panel of lines that have the mtDNA deletion between 0% to 99 and 100% homoplasmy. And you can see we have around 35 cell lines, all isogenic, all retinal pigmental epithelial cells. The only thing that's different in them is how much mutant mtDNA they carry. And with this cell line, we can start asking, what's the level of threshold? What's the cellular dysfunction? What's the metabolic dysfunction? Now, you might have been surprised because I said deletions are never found at homoplasmy, although these cells, many of which reach homoplasmy. So why is that possible? In fact, that's possible because cells in culture are grown in glucose media. We dump so much glucose on these cells that they do not do oxphos. They rely on glycolysis, and therefore they do not care how much mutant mtDNA they have. But you can unmask the defect if you shift these cells to galactose. And now what you get is any cell that has close to homoplasmy is completely dead in the water. These cells don't grow. Cells that have less than 75% homoplasmy are fine, and the threshold is 75. They're sitting around the plate for a while, they're not dying, and at some point, they pick up and magically start growing. And what happens at this level is that these cells are selecting against the deletion in galactose, and the 75% reach to lower than 75%, and these cells can grow. And that pretty much told us 75% heteroplasmy is the threshold above which all hell breaks loose. The accumulation of more of that of mtDNA leads to cellular dysfunction and really metabolic dysfunction. All oxphos subunits are gone, and metabolites like malate, citrate, aspartate are completely wiped out. Okay. So this was a nice characterization, but how does the nucleus respond? This is what I'm interested in. Does the nucleus know the mitochondria is in trouble? Does it know when there's complete collapse of the system, or does it know a little bit before? So to get insight into how the nucleus appreciate or realize what's happening in the mitochondrial DNA, we went back. It's not the clones anymore. This is the pool of cells. And we did single cell dogma-seq. This is a method that we developed, we adapted from others with a single cell innovation lab at MSK, involves doing a single cell attack-seq to read the mitochondrial DNA, together with single cell RNA-seq to read the nuclear DNA. So now you can, in each cell, ask how many mutant DNA mitochondrial DNA it has, and what's the transcriptional response like. And I want to make a long story short in the sense that we actually saw two modes of mitochondrial DNA damage sensing, mitochondrial DNA deletion sensing. The first is triggered by threshold. These are genes like mitobiogenesis genes, ECM degradation genes, which are stable. Then you get the transcription is flat. Then you get a certain level of heteroplasmy, which is here on the x-axis. And these genes are then expressed up or blocked. However, there is another group of nuclear genes, which are shown here, that are sensing heteroplasmy. So as you increase the heteroplasmy level, the RNA levels of these genes gradually go up or go down. And they're mostly genes in the calcium sensing pathway and some lung non-coding RNA. So basically, to summarize, we have a way, a tool that's actually easy. You can express it in any cell type you want to make any mitochondrial DNA deletion you want. We've engineered a panel of line that have the full spectrum of heteroplasmy, and to start to explore how these mtDNA deletions metabolically, molecularly, and cellularly affect cellular function. We believe 75% heteroplasmy is this threshold beyond which cells are completely collapsing. They're completely dysfunctional. But cells are also sensing this mtDNA deletion even before they reach this heteroplasmy. There's some counting mechanism that really takes notes of how many mutant DNA you have. And so ideally, if we know what's regulating this heteroplasmy sensing, can we bring in the heteroplasmy down and make sure these cells never get to that threshold? Okay, so I'm gonna acknowledge the people who did the work. The second part was led by a student in my lab, Yi Fu, who's now doing a postdoc on the West Coast. The work is carried out by Tata, a postdoc in my lab. This is a close collaboration between the lab of Craig Thompson at MSK, who teaches me everything I need to know about metabolism because I know nothing, and the group of Dana Pear, who does single-cell sequencing experiments and analysis. We have a lot of collaborators, Repair Therapeutics, who provide us with drugs, and many institutions and foundations that pay the bills at the end of the month. Most importantly, the one that's being attacked the most these days, NIH. And thank you for your attention. So I can take questions on any part, first or second. Yes. The non-coding sequence of the chromosome could be a source of extra chromosomal DNA. In principle, yes. But if it's non-coding, usually the ecDNA in the context of cancer gives you an advantage. And this is, let's say, the MYC, the FGFR. Because the cells need them, that you can amplify them easily on ecDNA. Now, people have seen microDNA and ecDNA of different parts of the genome, but I don't know if those give the survival advantage. Typically, they go there, they're degraded, and they're gone. Except for ribosomal DNA. Actually, in yeast, amplification of rDNA can happen on circus that then can go back into the genome. Yeah. Yes, 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 yes. Yeah, yeah. Source. Yeah, yeah. Upon damage, yes. Yeah, yeah. W- Yeah, it could be interesting to actually ask if MMEJ is regulating the abundance of these repetitive ecDNA. It could be interesting, actually, after irradiation. We have not looked. We should look. Yeah. Yes. So once this DNA accumulates to a certain level in the mitochondria, that mitochondria become dysfunctional. You lose membrane potential, and if you're on a seahorse, they can't respire as much. Why are they accumulating to that level? Why does the mitochondria not select against them? Why doesn't it send them to mitophagy? This is something we're interested in understanding. Yeah. Yeah. So it's actually, yeah, you're absolutely right. Different cells in our body have different mtDNA content. A muscle will have 5,000, a neuron will have 10,000, a fibroblast will have less than 1,000. And that copy number is set. So if you degrade mitochondrial DNA in a muscle that has 5,000 and you remove the source of degradation, it's going to go back to 5,000. That number is kind of set. And in worms, the accumulation of deletions lead to an increase in overall copy number because a deleted molecule is like a dead molecule. Surprisingly, though, in our cells, even when you get to 75-80% of the molecules being deleted, the copy number overall does not increase significantly. It goes by up to 1.2-fold at most. So I think there's going to be some differences between model organisms and human in terms of copy number control. Because ideally, if you increase the number of wild-type DNA, it doesn't matter how many mutants you have. You just make up by increasing the wild type. But it doesn't seem to be taking place in the human cells. It's a very interesting question. Which drug? Okay, I do not know. Okay, okay, I will write it. That's interesting. It's good. Yeah, mitochondrial DNA goes down. Yeah. Yeah, that drug blocks Pol gamma, which is a mitochondrial polymerase, as much as well. Yeah, yeah. Okay.
Threshold seems to be dependent on the nutrient conditions.
Yeah.
That provide any insight on the different tissue specificities of the
Yeah.
and are there any dietary ways?
That's $2 million question, because two questions, each one is $1 million question, right? The threshold, you asked the question, is the threshold that you see in epithelial cells going to be the same in muscle and tissue? So obviously, we made now we made this large scale deletion in iPS cells that we're differentiating. And I can tell you, I cannot make the differentiation. The process of differentiation is selecting against the deletion. So what we need to do is induce the deletion here and here and see what's the maximum level. Can we find things to mitigate the effect? You bet. This is what we're doing. And we're actually using those cells to do genetic screen and small molecule screen to ask which drugs or which genes will ameliorate the effect of the 75% deletion, which will either select for the deletion or make these cells basically rewire their metabolism so that they don't rely as much on that genome. Yeah, this is where we're going, basically. Thank you so much.